Good morning, everyone, and thank you for the invitation to present to you this morning. I'm Adrienne Gorny, so I'm the Research and Extension Nematologist at NC State. So I cover row crops, soybean, corn, cotton, sweet potatoes, and tobacco, but I can really help with questions about nematodes in other crops as well, if you might do peanuts or mixed vegetables. But today I wanted to share some research updates from my team regarding our work in nematode control in soybean. So we'll talk about our nematicide trials from this past year. Particularly, we've been doing work for management of the guava root knot nematode. This is Meloidogyne enterolobi. And we've also been doing some work in sting nematode, which is a highly damaging species of nematode. And then we'll go over some other management options. Nematodes are really best managed through multiple tactics, so including cultural controls with variety selection and nematicides. All of these working in combination tend to give us the best results. And then at the end, I'll give some research updates from some collaborative projects we've been doing for screening for guava root knot resistance in soybean varieties. So first though, when we think about nematodes that impact yield in soybean, these tend to be our usual suspects. Not all nematodes are created the same. Root knot and soybean cyst tend to be our most common and most impactful types of nematodes. However, other ones such as lesion nematode, sting, in certain parts of the state, reniform nematode can also be problematic. I've also been receiving some more calls and questions about stubby root nematode. So if this is one that you've seen on your nematode reports, I'd love to discuss that with you. Symptoms of nematodes in soybean. Again, these nematodes are soil-borne. They're microscopic and they're feeding on the plant roots. So during the season, this root damage may result in above-ground symptoms that are very similar to water stress, nutrient deficiency, other types of pathogens. So we tend to be looking for poor stands, yellowing, patchiness. You can see here, this is due to soybean cyst nematode. But these could be attributed to a lot of different things. It could be a nutrient deficiency. So to diagnose, we really have to look below ground, looking for evidence of root galling in the case of root knot nematode, and a soil sample to follow up. Soybean cyst nematode, again, this is a highly pathogenic one. It can produce similar symptoms, yellow patchy spots in the field. Soybean cyst nematode can be diagnosed if you dig up the plant and look very, very closely at the root. You may see these small white to yellow blobs essentially on the root. These are the body of the female nematode itself. They tend to fall off the root somewhat easily, so it is best to dig up the plant rather than pulling the plant out of the ground. You have to look very closely. These are going to be the size of about the tip of a pencil. That's about how big they are. All right, so moving into some of our nematicide work. Again, we were working both for management of the guava root knot nematode and management of sting nematode. And so this first trial was for the guava root knot, Meloidogyne enterolobi. And we did this trial down at our guava root knot nematode research site at the Border Belt Tobacco Station in Whiteville. We have a quarantined research site. It's about 8 acres in size. It's fenced in. We have a biocontainment protocol. It allows us to do very targeted research on this nematode in soybean and a variety of other crops without contaminating any surrounding area. So we're very fortunate to be able to have this and do important work down there. So for this trial, we're interested in evaluating several different rates of fluopyram for nematode control, and then some other treatments, what I'm calling alternative nematicides. Nematodes can be difficult to control, and so I'm interested in developing a data set to start exploring some of these more alternative types of nematicides. This was a small plot work. The plots were 4 rows wide by only about 40 feet long, and in that randomized design that Rod was talking about this morning, in order to capture the variability of the nematodes across our trial area. So we collected a variety of different data on this trial. We collected soil samples both at planting and at harvest, and from those samples we counted the number of guava root knot nematode that we found, and then collected yield data, stand counts, and some vigor ratings throughout the season. And this is what we found. So I'll start with this bar graph here on the left-hand side. So this is the effect of our treatments on the nematode counts at the harvest sampling. There was really no difference in the nematode counts from the early season sampling, so I won't be showing that graph, but they were fairly even across the board. But this graph here is for the harvest samples. The non-treated bar is here on the right-hand side. The vellum treatments going up in rate, and then the alternative nematicides here. So from this, there were no statistically significant differences among these treatments. However, numerically, it seemed that our crab meal that was incorporated in Asgard had the lowest nematode counts at harvest. But again, those weren't statistically significant, as very well described by Rod this morning. On the right-hand side here, this is the yield, the soybean yield at harvest, and this is expressed per the average of the plots. And again, there were no statistically significant yields among our treatments. The non-treated bar is this one here on the right. So this is actually not quite what I was expecting. I was expecting our vellum, the fluopyram treatments, to perform a little bit better than this in terms of reducing root knot nematode numbers and giving a bit more of a yield bump, but we did not see that this year. So the key takeaways from this trial was there were no statistically significant differences in terms of yield. We did see a reduction, again, of the guava root knot nematode with the vellum at the 6 fluid ounces there, and a couple of our other alternative nematicide. Just as a note, for this particular trial, we were using a product called Vellum Prime, which is labeled for vegetables and sweet potatoes. There's a new product just called Vellum, straight Vellum. That's the one for soybean. However, those two do have the same active ingredient, active nematicide in them. Moving to our next nematicide trial from 2023, and this one was for evaluating control for sting nematode. This is Bellona limus. This was done at the Central Crop Station with a very helpful crew there, and we had the same treatments applied at this trial that we had in the root knot trial. Again, small plot work, replicated and randomized. The same type of data were collected at this trial as well. So sting nematode in soybean and a variety of other row crops. Sting nematode is known as one that has a very low threshold. Even a few individuals in a nematode sample can indicate a high level of risk to that crop. So sting nematode tends to produce shortened, stubby roots. We see less lateral root growth, early termination of the roots. Sometimes the roots can tend to spread out very near the surface, but we don't get deeper tap roots forming, and very patchy distribution in the field, and pretty significant stunting when present. So sting nematode can impact corn, soybean, cotton, and a variety of vegetable crops. So these are the data for the sting nematode trial. Again, I'll start with this bar graph here on the left. This is for the number of sting nematodes on average at the late season harvest sampling. So again, this is the non-treated bar here on the right. So again, the lower the number here, the better. And all of our vellum treatments did numerically reduce the number of sting nematode on average, but there were no statistically significant differences among these treatments. This graph here is for the yield. Again, unfortunately, no statistically significant differences. However, that vellum prime at the 3 fluid ounces did have numerically the highest yield among these. So this is work that we're hoping to follow up this year with support from the Soybean Producers Association, who've helped support this nematicide work and future nematicide work, which we're very excited about. Yes, so we'll be doing work again a lot in management of guava root knot nematode and other types of pathogenic nematodes. After that nematicide work, I wanted to follow up with a few tips and tricks for sampling for nematodes and other management options. And this may actually be familiar to many of you, but I think a reminder is always helpful. When sampling for nematodes, we do recommend to collect these in the late summer or early fall. That's because the soil is warm, the nematodes are actively feeding, and it makes it easier for the NCDA nematode lab to find them and count them and identify them. But really, a soil sample at any time of the year is better than not collecting one at all. You can even collect one now. We just recommend to sample a little bit deeper than this recommended 8 inches deep. Collecting them on a zigzag or W-shaped pattern through the field. Again, this is to capture the variability of these nematodes that both Rod and Anders mentioned previously. As you can see here, nematodes do occur in these patches and hotspots, and you may not know where those patches or hotspots are in the field, so using this zigzag pattern can help increase the odds that you catch one of those hotspots. Sending them to the nematode assay lab as soon as possible after sampling. I know with everything else going on, it's sometimes difficult to get those in a timely way, but really trying not to delay too much on submitting those. This slide is to illustrate that sometimes it can actually be beneficial to sample from what we call the transition zone, in between healthy and plants that might be going down. So this sampling technique is very good for an in-season or during-season sampling. If there is the case where we have plants that are going down or might already be dead, these plants are not supporting the nematode populations like active healthy plants might be, or plants right in between. So if we sample from smack dab in the middle of a spot that's already dead, we may actually get a false negative report. So those numbers could actually be lower than what true populations might be, and we might then think that, oh, nematodes might not be the issue. But in this case, sampling from that transition zone right in between can help give a clearer picture in some cases. So once you have those samples in hand, keeping those in a cool location, but not putting them in the freezer. My team, we just use a standard drink cooler, pack them in there. It keeps them shaded and cool until we submit them. A special note here is that the NCDA actually runs a special molecular test or DNA-based test specifically for detection of the guava root knot nematode. However, this test can also distinguish other species of root knot nematode. So if you are interested in having this test performed, you'd have to select this last box here on the sample submission form. Just as a note that the lab does not save the soil in between samples. So if you send in one for the regular $3 test, but then decide later that you want the molecular test done, you'd have to send in a new soil sample. This slide here is to remind me, to remind you, that the NCDA lab actually updated their sample submission form a couple of years ago, mainly to include that molecular diagnosis option. So if you might have old sample submission forms around the office, you can still use those. They will still accept those, but they don't have that molecular diagnostic option. So these new sample submission forms you can find on their website, or I'm also happy to send you one too. The NCDA lab actually has two different types of nematode tests, the predictive test and the diagnostic test. Both of these tests are going to provide you with very good data about the types of nematodes that are present and their population counts. So both are excellent to use. But this predictive test is more to answer the question of, I'd like to plan for a great crop next year. This test is going to help me decide if a nematode management plan is needed, and if so, what kind of management options should we be looking at. This one is generally the one we suggest for those late summer or early fall sampling. This diagnostic test is to help answer the question, I'm pretty sure this crop has a nematode problem, but I'd like to rule it in or rule it out. This diagnostic test is the one we generally recommend for an in-season sampling. But when in doubt, I tend to go with the predictive test. But again, both will give you excellent information about the types and the counts of nematodes. I wanted to just offer a few tips about interpreting a nematode report here. So there's a lot of information packed into these reports, and we can tease out a few of them. So in this particular test, these are the types of nematodes they found. So they found root knot, soybean cyst, lesion, ring, spiral, and stunt. And so each of the lines obviously represents a different sample that was submitted. So under each nematode, there's two columns, the S column and the H column. The S column is just the number of nematodes that they found in that sample, the straight counts that they found in the lab. The H column is a hazard index. So based on this count, what is the level of hazard to the crop from this nematode? So for example, in sample 37 here, they found 10 root knot nematodes, which corresponded to a hazard index of about 1 to 15. We can figure out what this means by flipping over the report on the back and coming down here. So a hazard index of about 1 to 15 means predictive of a very low crop damage due to root knot nematode in this case. But if we look at the sample just below it, field 38 here, they found 1,000 root knot nematodes, and that prompted a hazard index of about 55 to 90. So that gives a moderate to very high predictive level of crop damage. So that can help interpret these nematode numbers here. In certain cases, there's no hazard index given for the associated nematode count. That is because generally there's not a good agreed upon threshold for that type of nematode in the crop. In that case, generally, the higher the number, the more concerned we should be about it. The lower the number, the less concerned. All of these numbers taken together are then summarized into an action code for that particular sample. So in sample 37 here, we had a very low level of root knot, low level of lesion, and a low level of stunt nematode. Taken together, this prompted an action code of A. We can again figure out what this means by looking at the back of the report. An action code of A is no expected harm due to nematodes in this crop production. However, for field 38 here, just below it, we found a high level of root knot nematodes, but nothing else. This prompted an action code of B, D, or E, which means possible damage. We may consider a nematicide treatment, use of a resistant variety or crop rotation. So this can help us to interpret these nematode reports of what types of nematodes and what their levels look like. In general, low levels of nematodes are not going to be an issue, but higher levels we want to manage effectively. Another thing is if you do have the molecular test done, the results of that are going to be tucked up here in this box where Dr. Wayman-Ye at the NCDA writes out what he found. So this was a molecular test. And he said that a high level of root knot nematode was detected, the molecular test was performed, and the species was identified as southern root knot nematode, Meloidegein incognita. So in this case, he did not find the guava root knot nematode in these samples. I'm always happy to help interpret these nematode reports if you have specific questions on a report or for a particular field. Here I compiled general threshold levels for our most impactful nematodes in soybean production. So we've got root knot, soybean cyst, lance, reniform, and sting nematode. So again, not all nematodes are created equal essentially. Some of them have lower thresholds and some have higher thresholds. So these are the numbers, assuming that this is a fall count, so samples taken in the fall, and assuming that the potential damage, assuming a relatively sandy soil. Samples taken in the spring will have lower threshold values than samples taken in the fall. That's mainly because we do see an attrition, a die-off of nematodes during the winter time. Just as a note, these tend to be ballpark threshold values. An actual potential damage to the crop is going to depend a lot upon certain varieties or cultivars grown, other practices. In the case of soybean cyst nematode, the HG type of the nematode you have in the soil, or a root knot species. But in general, these can help us decide if it's a low risk or a high risk to the crop. A couple of other reminders I just wanted to offer was about limiting the spread of nematodes. Nematodes are microscopic. They're not very fast movers. A lot of estimates say that they can only move about 5 feet maximum on their own per year. So long distance movement is unfortunately facilitated by people. But sanitation, cleaning tractors and other equipment between locations can help limit this spread. Particularly for the guava root knot nematode, we recommend a 10% bleach solution. But really just washing off that infected soil, getting that knocked off before moving to the next location is going to reduce the risk of spreading nematodes and having them establish in new areas. Starting clean and staying clean is the best preventative measure for nematode control. I included this slide because I've had some really excellent conversations recently, questions about the impact of tillage on nematode populations. So questions about certain tillage practices and if that can be used as a nematode control. So unfortunately, few studies have been performed for looking at the effect of tillage on nematodes, but of those that have been performed, for root knot and lesion nematode, there was no consistent influence or impact of a tillage program on nematode management. For a few of the soybean cyst nematode trials, Some of these indicated that a no-till or reduced-till systems had lower SCN populations, but the effect wasn't very strong, and additional research into this is needed before we can fully recommend this as an option. It is known, though, that soil texture, temperature, and cropping rotation have a greater impact on nematode populations than tillage. But again, just as a note that tillage equipment can move nematodes to new locations. So sanitation, again, is important here. All right, if I'm doing okay on time, I'd like to just finish off the talk with some research updates from our greenhouses on the NC State campus. So this is some work that does not necessarily have immediate impact in the field. But we are involved in collaborations for trying to identify new sources of resistance in soybean to the guava root knot nematode. Currently, all commercial varieties of soybean are susceptible to the guava root knot nematode, even those labeled as resistant to nematodes. Those labeled as resistant to root knot nematode will be resistant to southern root knot nematode, which is our most common species of root knot across the state. So to address this, we've been working with some soybean breeders to evaluate for new sources of resistance. So eventually that down the road, we can develop a resistant variety to the guava root knot nematode. And this was a research project undertaken by Tanner Schwartz, who was a PhD student in the lab, who recently graduated and received funding support graciously by the Soybean Producers Association. So what Tanner did was he screened 44 commercial soybean, this is glycine max, and 77 wild soybean, glycine soja, in the greenhouse for resistance to the nematode. So this is just a snapshot of what it looked like in the greenhouse. Tanner had everything very organized in there. He evaluated all of these different lines of soybean, inoculated the plants, allowed the plants to grow, and then came back about 60 days later and evaluated whether or not that plant was infected, and if so, to what degree it was infected. And this is what he found. So this is a little bit busy here, but I'll start here on the right-hand side. So none of the commercial soybean lines that Tanner tested were resistant to guava root knot nematode. All of these were susceptible, which means that we need to, at this time, manage the guava root knot nematode through nematicides and rotational crops in soybean. However, he did find some promising results in the wild soybean. So only five out of the 77 that he screened did have resistance to the guava root knot nematode, and it was very strong resistance too. So all these numbers are just a summary of the wild soybean responses. So these different rows represent the different lines of soybean. And we tend to look at these RF factor, which is the reproductive factor of the nematode. All of these numbers are well below 1, which we use 1 as the cutoff for susceptible or resistant to these nematodes. So this was promising results that we were able to find some resistance in wild soybean to this nematode. And we've been working again with the soybean breeder in Raleigh. We're going to continue this project, try to identify the genes or the loci in the genome that's responsible for this resistance so we can better understand it, better characterize it, and then hopefully down the road we may be able to incorporate this resistance into soybean. We do have a new graduate student, Sarah, who will be continuing on this work. So hopefully next year we'll be able to provide some updates to this. Just to illustrate what this looked like in the greenhouse, so here on the left we have the wild soybeans, and here on the right we have the cultivated soybeans. The resistant line was very clean, no galling, and heavily galled in the susceptible varieties. So with that, I'll just say thank you very much again for having me at your meeting, and if there's any questions, I'd be glad to take those.
Have you done any studies to see if, like, if you put the vellum on the soybean crop this year, and then the next year in a rotation with sweet potatoes or whatever, if it knocks back the populations and helps with the next crop?
That's a great question. So essentially, if there's a carryover or residual effect to the next crop from an application, say in soybeans in this crop, we have not specifically looked at that. In general, we see that by the end of the season, the nematode populations have rebounded from that early season nematicide application. That early season nematicide application, whether it be a fumigant or a non-fumigant nematicide, is very good at knocking back the nematodes during that critical early growth stage, and that's what we're looking for. Generally, by the end of the season, once the soils get very warm and the nematodes are active, they tend to rebound to their previous levels. So I would not expect that there would be a benefit to the next season's crop in that case. Yep.
How much pressure was the site we had nursery?
That's great. So the question is, how much pressure was there at the guava site? At last year when we did this, we had what I would call moderate pressure. We were looking at 200 to 300 nematodes per 100 grams of soil, which I consider a moderate level. It will build up over time. Last year was the second year that we've done research trials at that site. So this year will be the third year. Yep.
So I guess y'all are going to try to do whatever we can to enhance that population.
Yes, so at that research site, we really actually do want a good population so that we can look at the effect of these nematicides.
The resistant soybean varieties, you used, I think, an Astro 57 something.
Yes.
You test, that's a pretty high rated nematode variety, correct?
I believe it is, yes. So that would be resistant to southern root knot nematode, and in this case it was guava root knot nematode that we were testing, and it would have no resistance to guava. Correct, unfortunately. We have done some testing with aldicarb, not specifically for guava root knot, but for other types of nematodes. It's been a mixed bag, and I think it has to do a lot with the conditions at application. Sufficient moisture, temperature, things like that leads to best success with that product.